The human microbiome, those bacteria, protists, fungi, and viruses which naturally occur in and on the human body, includes so many organisms that the number of microbial cells associated with the human body actually outnumbers the number of human cells in the human body. The effects of the species in the microbiome, their relative abundance, changes in the microbiome, and their activities do affect human health. And a growing field of medicine is to investigate the relationship of the microbiome and potential health versus disease. For example, changes in the microbiome have been linked to the onset of type 2 diabetes. Might the microbiome be linked to the onset of type 1 diabetes? Although it should be kept in mind that human and mice are obviously different, and thus studies in mice may not be entirely applicable to human disease studies. Nevertheless, there are mice strains which develop diabetes, such as the NOD strain, which are used to model human diabetes. In these rodent studies, the development of diabetes can be correlated with changes in the species of the microbiome which exist and their relative diversity. So for example, some species are known to be less abundant, others more abundant. Some alterations in the number of nutrients processed by the microbes of the intestinal flora. These can be correlated with the incidence of diabetes in these rodents, comparing rodents which have diabetes versus those which do not. These changes in the microbiome can heighten the inflammatory state of the digestive organs, including the pancreas, which could certainly affect the progression of inflammatory reactions in diabetes. It has also been noticed that antibiotic use can affect diabetes incidence in rodents, and that some bacterial strains seem to offer protection from diabetes. Diabetes in humans does have some similarities to these observations in mice, including there's the decrease in the abundance of firmicutes in those who develop diabetes, and the relative increase in the abundance of bacteroidetes. There is also the increase in the inflammatory state overall in digestive organs. In modern societies, the incidence of type 1 diabetes is rising. Also in modern societies, there have been changes in overall hygiene, disease incidence, including the frequency of diseases affecting the gastrointestinal tract in childhood, and other societal aspects which have changed the human microbiome and the conditions in which the immune system of the gut matures. Changes in the microbiome have been observed in children with diabetes compared to children without diabetes, and changes in the microbiome have preceded the observed change to diabetes in children which were genetically predisposed. In early childhood, there are three phenomena which are occurring simultaneously. The gut microbiome is transitioning from its composition in infancy to its adult composition. At this time, the immune system is maturing, in part due to interactions between immune cells and microbes in the environment. And this is also the time when those who develop onset of diabetes during childhood are observing antibodies against autoantigens such as insulin, which arise before the diabetes occurs. Just because three events are correlated does not mean one causes the other, but there might be a more direct interaction between these three variables.